Hi everyone, this is Chapter 5. We're looking at the structure and function of the plasma membrane. To start, the plasma membrane is often described with what's called the fluid mosaic model. Fluid because all of those phospholipids, remember a phospholipid are these cute little guys with the phosphate head and the two little fatty acid tails? Well, when you add a bunch of those together, they kind of are able to move around back and forth and kind of slide up and down on one another. So it makes the plasma membrane kind of fluid. And then the reason that we call it mosaic is, perhaps you don't know what this word is. A mosaic is something that's kind of a decoration made out of little pieces of something. So oftentimes it's tile, and you can see that there's a bunch of little pieces of cut up tile that arranged in a certain way make kind of an interesting shape. Well, in that respect, the plasma membrane looks something like a mosaic. So what you're seeing here, this is actually an electron micrograph showing the outside of the plasma membrane. And over here, this is where it's kind of the bird's eye view of a cell. And you're seeing here, this is where the two layers of the bilayer are present. Well, if you peel one of those layers away, this is kind of what it looks like over here. And you can see that there's a bunch of kind of stuff sandwiched in between the two layers of the phospholipid bilayer. Again, remember, phospholipid bilayer means if we're looking at the side of it, it would look something like this, right? And so when you're looking at this picture, you're looking at the top down of the outside of the plasma membrane, and then if you strip away one of those bilayers and you're just looking at what's in the middle here, that's what you're seeing speckled around here. All that to say, the plasma membrane is far from just, sometimes I make the mistake of saying it's just a film of oil, which in one respect it is kind of a film of oil, but in another sense it's much more than that, because embedded in the hydrophobic fatty acid tails of phospholipids are tons of these little proteins. Okay, so that's why we call it a mosaic, and we'll become more acquainted with those proteins in a little bit. Here's kind of a picture that shows the story. Here's our two layers of phospholipids. Again, these can kind of move up and down against one another, so in that sense it's kind of fluid. And then each of these proteins is kind of embedded in that membrane to create kind of the mosaic appearance. There's an interesting animation, and this comes from what's called the Life of a Cell. And this was put out by, I think, Harvard University or something like that a couple of years ago. It's a big 8-minute long narrated production, but I've just included a small snippet of it here so that you can see kind of they've got some interesting animations depicting what the plasma membrane is like. So you can see all of these phospholipids are kind of moving back and forth from one another. You might find a whole bunch of different types of proteins moving in and throughout the membrane. There's some that are embedded in the membrane, such as this one. Some are directly outside of the membrane, kind of in this extracellular space. And then as we descend down into the inside of the cell, this is kind of what the underside would look like. And then you've got, stuff attaching into the plasma membrane and different proteins that are anchored into that hydrophobic part of the membrane. Some proteins are just attached kind of to the surface of the membrane. So it's really just a very complex structure, the plasma membrane. Okay, it's got a variety of functions of it, right? So here's just for reference sake, here's a electron micrograph, so a picture taken with an electron microscope of a cell, and this is kind of showing the cross section of it. So if you were to just take a thin slice of it, this thing that I've kind of drawn around the plasma membrane is the thing that surrounds the cell, but it also is something that surrounds a lot of the organelles too. So like here we have mitochondria. They're each going to have their own little plasma membrane as well. And the nature of the plasma membrane surrounding the cell could be similar to that of the one that's surrounding the mitochondria. Remember, the nucleus has a nuclear membrane, another plasma membrane surrounding it. Things like the Golgi apparatus, these all have little plasma membranes around them. So there's a lot of plasma membrane in a cell. One of the biggest things that we usually think of, though, is that it isolates the cell from the outside environment. Okay, so right here, this is inside a cell, and inside all of this stuff is considered living, but as soon as you go outside of the cell, then suddenly this space is stuff that's not considered living. And so the thing that's required for life is to maintain a pretty constant environment. And so when you have a barrier like the plasma membrane, it means that all the stuff happening here inside the cell can be kind of held at a constant temperature and a constant consistency despite any kind of changes in the outside environment. So that's a big function of the plasma membrane. It's also going to allow certain things to come into the cell and also allow certain things to exit the cell, so it can determine what comes in and what comes out. So in that sense, it regulates the exchange of important things into and out of the cell. It can also communicate with other cells. So that plasma membrane, that thing that's on the outside of the cell, it can have little receptors and things on the surface, which we'll talk about later, and that can help to either attach to other cells, or it can be used as a means to tell other cells a little bit about the cell. So for example, this particular cell right here, perhaps this is an example of a skin cell or something, and as a skin cell it puts a certain array of little pieces on the outside that will tell other parts of the body, hey, I'm a skin cell. Okay, so that can help communicate that type of thing with other cells. And then, like I said, it can create attachments between cells. So for example, your skin, as long as we're on the example of skin, your skin has a whole bunch of cells. So this is kind of the side view of your skin. This is like the surface of your skin, and then this is deeper as you go deeper into your skin. And you can see there's just a whole bunch of cells all attached to one another. And the way that they attach to one another is that they're bound through these little adhesion proteins that are on the surface of the plasma membrane. So they allow one cell to stick to another cell. And then one of the last things here about the plasma membrane is it's going to be a place where there are a lot of biochemical reactions that happen. And that's because on the surface and embedded in those enzymes are embedded in those membranes are enzymes. And enzymes, remember, are the things, they're proteins, and they're proteins that are going to help regulate chemical reactions. They can either speed up chemical reactions, or they can turn certain chemical reactions off and keep them from happening. So those are some of the things that a plasma membrane does. Excuse me. Let's talk a little bit about what is inside of a plasma membrane. The first is that it's our infamous phospholipid bilayer. Okay, so remember, at this point you should be able to know what a phospholipid is. Phospholipid is something with a phosphate head on it, and it's got two fatty acid tails. It looks something like this. Okay, so here's that phosphate group, and then there's a molecule of glycerol, and onto that the phosphate group occupies one part of the molecule, and then the other two parts of the glycerol molecule can attach to two fatty acids. And the biggest thing to remember about this molecule is that the phosphate head is called hydrophilic, and the fatty acid tail is called hydrophobic. Remember, hydrophilic is anything that's like water. Water has slight electrical charges on it because of the difference in how electrons are shared between oxygen and hydrogen. And so this thing right here, this phosphate group, you can see it's got a negative charge on there. And so charged type stuff likes to be with charged type stuff. So that's why this part wants to be next to water. The hydrophobic part is the part that's doesn't have any charges on it. This is just a bunch of hydrocarbons, and hydrocarbons, as we discussed in chapter 4, I believe, or chapter 3, are just a bunch of carbons loaded with hydrogens. Okay, so there's no unequal sharing of electrons in these molecules, so there is no charges, and because of that, they do not like to associate with water. One thing that's kind of interesting about the fatty acids that you would find on a phospholipid bilayer. Sometimes the fatty acids can be saturated, as is the case right here, where you see there's no double bonds between any of these carbons. Whereas over here, you can see that there's a double bond here between two carbons. Remember, each one of these points is it's assumed that there's a carbon there. Okay, so this bond here, these two bonds between these two carbons, that kinks the molecule. So that's why you see there's a bend here on this particular fatty acid. And so organisms have a way of kind of increasing or decreasing how fluid this whole layer of phospholipids is. If they insert a bunch of unsaturated fatty acids into the phospholipids, then all of these kinks make it so that the phospholipids can't pack as tightly as perhaps this picture shows, and so it makes the membrane a little bit more fluid. This is good in colder regions or cold climates. So for example, fish that have to survive in freezing, cold water within lakes during the winter, they actually will increase how many unsaturated fatty acids are in their phospholipids in order to make sure that those membranes are still slightly fluid even when it gets really, really cold. Whereas us, we usually keep a pretty much constant body temperature, and we're able to, modify our surroundings, like, heating up our homes and putting on jackets and things like that to maintain that temperature. So we can just mostly have saturated fatty acids in our phospholipids. So the phospholipid bilayer, again, it's important because it's got a hydrophilic and hydrophobic region. All of this stuff here, the fatty acids, this is all hydrophobic, so this is kind of oily. It's made up of a bunch of fatty acids, and then all of these little balls here along the surface, those are hydrophilic, meaning they're attracted to water. And keep in mind, there's a bunch of water on the outside of the cell and a bunch of water on the inside of the cell, and so that's why all of these hydrophilic heads are going to point this way towards the water, and all of these hydrophilic heads are going to point this way towards the water, and then that's what makes it so that all the fatty acid tails face one another. Also in the membrane is cholesterol, and cholesterol, its big job is to stiffen the membrane. And cholesterol, recall, is made of a steroid, which means 4 carbon rings that are kind of fused together. So these little things right here are cholesterol, and they are going to be a little bit more rigid than these fatty acid tails, and so when you insert those into the membrane, they help give it some strength. 40% of the plasma membrane is made up of lipid type stuff. So cholesterol is a lipid type substance, and then phospholipids, as the name suggests, are lipids. And so if you take a look at all of the cholesterol and all the phospholipids, it makes up about 40% of the plasma membrane. Proteins are the other thing that make up a large percentage of the membrane. And there's 2 types of proteins that you might find in the membrane. There's what are called integral proteins. And as this name suggests, these are integrated into the membrane. So they're stuck, or they're embedded in it. So like this one right here would be an integral protein because you can see it's stuck into the middle of this hydrophobic space. Here's another one. Here's another one. Here's another one. Okay, so those are all integral proteins. And the way that a protein is determined to be integral is whether it has hydrophobic regions. And remember that a protein is just a big long chain of amino acids, right? And if there's a long chain of amino acids and there's some segment in that long chain, let's say this chain goes on forever and forever and forever. Okay, let's say there's some region of these amino acids that are all hydrophobic. The chemistry of those amino acids makes them hydrophobic, whereas all these, some of the other ones are hydrophilic. Well, then this hydrophobic region can actually get inserted into the hydrophobic part of the membrane, which is this whole part. Okay, so that can happen. Other times you might have several regions on a long chain of amino acids that are hydrophobic. If that's the case, then the long chain of amino acids will go through the membrane once, and then go through again, and then go through again. And so then, like in this example, you have 3 regions of amino acids that are collectively pretty hydrophobic. So then it passes through the membrane 3 times. You could even have something super complicated like this that passes through the membrane a whole bunch of times. Okay, so and then also these regions that pass through the membrane, they can either be alpha helices, or beta pleated sheets. But in either case, if something is going to be embedded into the membrane, it's going to probably have a large series of amino acids in its chain of amino acids that are hydrophobic. Next up, excuse me, are the peripheral proteins. Peripheral proteins means that they don't actually get inserted into the membrane. They just kind of sit on one side of it. So like this would be a good example here where you see it's just kind of nestled up against one edge. It doesn't actually pass really too far into the hydrophobic area. If you look at all of the protein, okay, so that's this stuff, that's this stuff, that's this stuff, that's this stuff. If you look at all of that, count it up, it makes up about 50% of the volume and the weight of the, I think it's the volume of the plasma membrane. Then there's one last category of biological molecules that you would find in the membrane, and that is the carbohydrates. And what they do is they're always found on the exterior of the plasma membrane, okay? and they're generally bound to proteins and to lipids. So, for example, right here we see a carbohydrate. This is a polysaccharide because it's made up of, you can see, several sugars all linked together. And that is attached to a protein, this little blue thing, so we call it a glycoprotein. Whereas you could also have a whole bunch of, a polysaccharide or a couple of sugars attached to a lipid, so one of these fatty acid tails or cholesterol or something, and then we call it a glycolipid. So this word glyco is usually referring to sugars of some kind or polysaccharides of some kind. So glycoproteins and glycolipids. Now the purpose of these sugars mostly is for cell recognition. Okay, so these are the markers, the specific things that a cell will put on its surface of its membrane to tell other things what it is. So for example, my pancreas cells, the cells in my pancreas, are going to have a series of these different types of sugars that they display, which will tell the body, hey, I'm a pancreas cell. Red blood cells, the blood cells in your blood that carry oxygen, they're going to have a series of these special glycoproteins and glycolipids that tell the body, hey, I'm a red blood cell. So it allows your body to kind of differentiate between different cells, and that would be the case too with all other types of organisms, not just you and I as humans. Because all of this sugar is found kind of on the outside, oh, I'll back up. Here's an interesting picture showing the virus HIV. So this is the human immunodeficiency virus, HIV. And one of the reasons why it is able to invade a host is because it attacks these glycoproteins that are found on white blood cells. So specifically, this one is called a CD4 molecule, and it's found on lymphocytes. It's a special kind of white blood cell. And HIV can seek out and attach to those cell receptors, and then because of that, it allows HIV to enter the cell. Normally, the white blood cells have these receptors to tell the body, hey, I'm a white blood cell and I'm here to help, because white blood cells help us fight off infections and things like that. But in this case, it's kind of a bad thing because there's a virus out there that knows how to recognize those cells and unfortunately destroy them. That's an example of where carbohydrates are found kind of in pathology or diseases of the organisms. Together, all of this sugar that's found on the exterior of the cell, it forms kind of almost like a sugary layer, which we call the glycocalyx. And there's also going to be some some sugar molecules kind of in the extracellular matrix just surrounding the cell as well. What the glycocalyx does is it is going to attract water to the cell, because sugars are kind of hydrophilic, and so water is kind of drawn to them. So it almost creates it kind of pulls water in so that there's a thin layer of water just outside the cell, which is good because cells need water in order to use as a medium to let stuff leave and then enter the cell. That stuff has to kind of diffuse or travel through water. So there should be a nice little kind of layer of water surrounding the cells. Okay, in your book there is a chapter 5 or table 5.1, which is going to review a lot of these components and where they're found. So if you would like to do a little review, I would recommend that you kind of just peruse that table, maybe kind of cover some of these things and quiz yourself. All right, so the first review question that I have for you is go ahead and try sketching. Make a little sketch of a plasma membrane. See if you can remember in your mind's eye what the arrangement of this structure looks like. How the phospholipids look like, how they're arranged in a phospholipid bilayer. In the phospholipids, go ahead and label which one is the hydrophilic part and which is the hydrophobic part. Maybe draw an integral protein, a peripheral protein, a channel protein. Don't worry about this, I haven't talked about these yet. And then a glycoprotein and a cholesterol molecule. Go ahead and pause the video and see if you can come up with a sketch. Alright, so what your sketch should look like should certainly have a bunch of phospholipids. And again, phospholipids look like this. They have a cute little ball head with little fatty acid tails. Okay. So a phospholipid bilayer is one that has two layers of phospholipids. Here is the first layer. Here is the second layer, and you should have drawn them so that the fatty acid tails face one another, and then the phosphate heads face the outside. And so this would be the hydrophilic part, and this would be the hydrophobic part. Also, integral membrane protein would be one that spans the whole length of the membrane, whereas a peripheral protein would be one that is just on the surface. So this one right here would be integral. This one right here would be peripheral. And then what else do we have? Glycoprotein. So if I put a protein in here and I attach some sugar molecules onto it, making a, these sugar molecules can either be a long chain or it could be branched, or maybe it's just one molecule, maybe two, but it doesn't really matter. If you have sugar molecules on a protein, we call it a glycoprotein. And then lastly, we have cholesterol. So cholesterol is a 4 fused rings attached to one another, and that would be inside the hydrophobic part of the membrane. Okay, so if you felt like you could do that, then you're at least thinking about the plasma membrane correctly. All right, we're going to get a little bit more complex now. We're going to talk about membrane transport, how things get into and out of a cell, and this is actually a really important biological process that happens in the body in any sort of cell. So there are a variety of types of ways that the cell can actually get stuff into it or get rid of stuff by exporting it out of the cell. And one of the simplest forms is diffusion. Diffusion is when you have something moving from a high concentration to a low concentration. That's kind of the most fundamental definition. Okay. And what we mean by concentration, this word just basically refers to the amount of stuff. Oh, can't spell today apparently. I can't really spell any day. You've probably found that out by now. Amount of stuff per given volume. So let's say I have some given volume, a space here and a space right here. And let's say in this space I have tons and tons and tons and tons and tons of stuff. This could be sugars or proteins or ions, particles, whatever, or just a little bit of stuff here. We would say that this is more concentrated and this is less concentrated, just because in the same region of space there's much more stuff here. Well, generally what happens then is that when you have a whole bunch of particles close together next to each other, they kind of they bump into each other, because in reality these things are actually moving back and forth. And as they're moving back and forth, they're colliding with one another. And as they collide, they slowly are going to start traveling further and further and further away from one another, so they spread out. And so in that sense, right here, this is a high concentration. By the way that you can write this is use these brackets. These little brackets refer to concentration. And then over here, this is a low concentration. And so you can see that the particles are moving from high to low. Now eventually, as they spread out, they're going to start spreading out, and now we have a concentration gradient. A concentration gradient is any difference in concentration between one space and another. So like over here, if these are continually spreading out, and right here I have only just a couple particles, that's a really low concentration, whereas here this is a really high concentration, and this in the middle is like a somewhat medium concentration. So a concentration gradient gets set up where there's a continual kind of spectrum of varying concentrations going from high to low. Well, when we're talking about diffusion, things always go down a concentration gradient. And when we say going down, all that means is that particles like to go from being in a crowded spot to a less crowded spot. So they go from a high concentration to a low concentration. Okay? If that's what's happening, it's called diffusion. So here's kind of an example of what that might look like for membrane transport. Let's say that this is the outside of the cell, and let's say that this is the inside of the cell. And here is some substance, and here's our plasma membrane. Well, right here, there's a lot of particles in a given amount of space, and there's none here. So this is a very low concentration because there are no particles there. This is a very high concentration. And so there's going to be a tendency for these things, as they spread out, to move from their high concentration into a place that's less crowded, the lower concentration, right? So that's going to push these molecules this way, going from high to low. And so over time, you're going to get the movement of some of these things into this direction. Now we are starting to change it a little bit because this is still a high concentration relative to this, but this concentration is a little bit less than this because, you know, some of our particles have been able to spread out some more. But we still have high to low, and so this will keep happening until generally what happens is that the particles reach what's called equilibrium, meaning that the amount of particles in a given space on the outside and the amount of particles in a space on the inside, these are the same. Okay, once this happens, there isn't going to be any net movement of particles because there's going to be no tendency for them to move from point A to point B. But let's say I add some more of these particles over here, then suddenly I have a concentration gradient again because this is slightly higher than this, and so that's going to continue to push some molecules that way. Alternatively, if I wanted to add some more over here, then that would make this the high concentration, and then they would flow this way. So hopefully that makes sense. That's an important thing to understand in this chapter, is what diffusion is and kind of a little bit about how it works. There's a couple of things that will affect how fast things diffuse. So the different factors which will affect the rate of diffusion. And the first is the solubility. Okay, so when we're talking about specifically about transport across a cell membrane, plasma membrane, one of the really important things to consider is, can the solute dissolve in lipid? Okay, I guess a better way to say this is, forget I ever said that. A better way to say this is this. Is it hydrophobic? Because if it is hydrophobic, that means it will dissolve better. Okay. If something is hydrophobic, it is able to pass through this membrane much easier. Because remember, a good chunk of the membrane, I mean, if you look at the width of this membrane, most of it is the hydrophobic part. And so something that's hydrophobic, it would have to kind of sneak past the hydrophilic heads. You know, hydrophilic stuff doesn't really like to welcome hydrophobic stuff, but assuming that it can sneak between those heads, then it's able to diffuse right through this hydrophobic part of the membrane. So stuff that's hydrophobic can diffuse across the membrane about a gazillion times easier than something that's hydrophilic. Okay. So generally when we're thinking about what type of things can just go right through the membrane, it's usually something that's hydrophobic. Also, what could affect it is temperature. So if I heat everything up, that means these molecules are going to be crashing into one another more frequently. And if they're crashing into each other more frequently, then that's going to increase the rate at which they spread out. Another thing could be the mass of the molecule. The bigger the molecule, the more sluggish it is to move. So the lighter the molecule, the more agile, and the more it can kind of move and then diffuse. The concentration strength of the concentration gradient, like here, this is a high concentration and this is a low concentration, so there will be diffusion, but it's going to happen slower than, let's say, this picture where the concentration difference is much greater, because this is much higher concentration and a much lower concentration than, let's say, this picture. So, you know, this would happen faster, this would not happen as fast, and so that's determined by the difference in concentrations from point A to point B. Also the solvent density, so what's the density of this fluid in which the particles are moving through. If it's really dense, it's like a jello, then it's harder for stuff to diffuse through it. If it's more liquidy like water, then it can diffuse a little bit easier. And then also the last thing would be the travel distance. Okay, so if I've got to go from way up here to way down here, that's going to take a lot longer than just from going here to here. All right, so that's a little bit about diffusion. Now you can break diffusion into two different types of diffusion. They're both diffusion, it's just one happens unaided and one happens aided. So the facilitated is the one that requires protein to happen. Whereas simple diffusion just happens, nothing is required. And so simple diffusion only generally happens for two types of molecules. Any molecule that's super, super small, or any molecule that is hydrophobic. And again, for that very reason of, if here's my phospholipid bilayer, a small sample of it anyway, in order to make it through the fatty acid hydrophobic region of that membrane, it's got to be hydrophobic. But if something is really, really small and it's not hydrophobic, it still might be able to quick sneak through in between these lipids just because it's so small. So for example, oxygen, the molecule oxygen that you and I breathe is two molecules of oxygen bound together, two atoms of oxygen bound together. That is not hydrophobic, it's sorry, it's a bad example. Another example would maybe be like, well, never mind. We'll just leave it at that. Okay, so it's got to be small or it's got to be hydrophobic in order to get through the membrane. Like sugars, like a monosaccharide like glucose is relatively small, but because it's hydrophilic, it doesn't quite sneak through the membrane. And same with like an amino acid. An amino acid is a little bit bigger, but it is hydrophilic, so it doesn't just diffuse right through the membrane. But clearly, we have amino acids and sugars get into and out of the cell. So how is it that they get into and out of the cell? Well, that's where we get into facilitated diffusion. Assuming that something is either large or perhaps it's hydrophilic, it can still diffuse into a cell. It just needs a little something extra. It requires protein. And the two different types of protein are the following. One is a channel protein. And what it is it's basically just a big hollow tube that's made by a protein. So the protein folds in on itself and it creates kind of this open passageway. And when that happens, then suddenly anything that's hydrophilic, such as a sugar or an amino acid, can just diffuse right on through, assuming that there is a concentration gradient. There's a high concentration here and a low concentration there. Okay, so that's a channel protein. And then another type of protein that you might find in facilitated diffusion is a carrier protein. And those are kind of similar to a channel protein. It's just a little bit different in that it requires a change of shape. So we'll just say a shape change. So in this scenario, what happens is that the particle will actually bind to the protein and the protein will change its shape. And after it changes, then it's suddenly able to be kind of spit out, as this picture is showing you. Okay. So stuff can't just pass right through. It has to go and bind, change shape, and then finally it can make it to the other side. Now, carrier proteins and also channel proteins. Channel proteins are often a certain size that only allow certain things in. So some channels will only allow like glucose in. Other channels will only allow sodium ions in. Another channel might only allow potassium ions in. So some of these are pretty specific, and then also carrier proteins, sometimes something might have to bind to it, and whether it can bind or not, it depends on the chemistry of the molecule. So there are some carrier proteins for maybe glucose, other carrier proteins for something else. So all that to say, whether or not there are certain proteins in the membrane can determine whether or not something can pass in, across, or out of the membrane. Okay, so because of that, the cell is often considered to have this characteristic known as selective permeability. What that means is that permeability refers to the ability of some substance to pass through some sort of barrier. Okay, so like if I have some barrier here and something can easily pass through it, it's considered permeable. Whereas if I have some sort of barrier and something is not able to pass through it, then it's not permeable. So that's what permeable means. And the membranes in cells are selectively permeable, meaning they can determine how permeable and what kind of permeability the membrane has. So maybe the cell is going to be really permeable to glucose but not fructose, or maybe the cell membrane is going to be really permeable to chlorine ions but not sodium ions. the cell has that ability kind of to moderate that, and it's not such that anything can just pass into and out of a cell, but some things can, and other things can sometimes, but other times not. So that's what we refer to as selective permeability. All right, one last form of diffusion is called osmosis, and osmosis is a special kind of diffusion that happens with water molecules. So when you see osmosis, I want you to think of water. And what happens in this case is usually this is the diffusion of water across a semi-permeable membrane, a membrane that is permeable just to water, or at least mostly to water, and not anything else. And that's fully permeable to water, but not anything else. Okay, so here's an example to kind of illustrate what happens in osmosis. Here we have a semi-permeable membrane. Okay, and this semi-permeable membrane will not allow these big purple solutes to pass through. Remember, the solute is the thing that's dissolved into the solvent, which is the liquid part. Okay, so this is solute, and then the liquid part right here, this is all this stuff. This is the solvent. Together we call it a solution. All right. So the solute in this example is not able to pass through this membrane, but the water can. Okay, so keep that in mind. One thing you need to know about water is, this is what I like to say about water, is that it's a party animal. Okay, and specifically it likes to go to whatever solute party has the most people. Okay, so if water is searching around, let's say, and there's one region that has a lot of solutes, it's a high concentration of solute, meaning there's a lot of solute per given area, and there's another region which does not have a lot of solute, water always tends to want to go to the place with the more solute. It's the solute party animal. Okay, all right, so here's two parties, a party here and a party here. There's much more solute partiers right here, right? So water that's here is going to always want to go to this party, and I'll explain why that is in a minute. But if this was this scenario, water would go into this party, and what happens is that it's going to flood that region of the beaker until you have an equal concentration of solute on either side. So there's the concentration of solute here is about the same as the concentration of solute here, because remember, in a given amount of space, let's say the given amount of space is these blocks, there's about the same amount of solute here and the same amount of solute here. So these have an equal concentration now. Even though there's a lot more substance over here, they still have the same concentration. So that's what happens. If you were to do this experiment, water is drawn in, and even though gravity wants to push these layers of water down so that they're the same level, the tendency for water to want to diffuse into the place where more solute is going to mean that the water actually raises and kind of defies gravity, so to speak. Now, why this happens? It all has to do with diffusion. Now you might say, well, isn't there a higher concentration of particles here than here? So isn't diffusion going to occur this way? Well, certainly the solutes are going to want to diffuse that way, so these little purple balls would want to diffuse that way. But take a look at the water molecules, right? Because there is so much space occupied by the solute here, you know, let's say that all the water molecules are in this kind of pink fluid here. There is a lot more water molecules on this side of the beaker, right? Because there's not as much space taken up by the solutes, right? So you've got tons and tons of space for water. Well, over here there's not as much water because it can't fit between all of these particles as well. So in the solute terms, there's more higher concentration of solute here, but when you're talking strictly about water molecules and compare the two sides of the beaker, there's actually a high concentration of water and then you also have a lower concentration of water over on this side. And so water just wants to diffuse simply, right? It just goes from its high concentration to its low concentration. So that is why osmosis does occur, and if you did this scenario, it could even defy gravity, and the water molecules will diffuse over here until the concentration of water molecules on this side is the same as the concentration of water molecules in this side. Okay. Now, this gets us to an important biological topic known as tonicity of solutions. And the tonicity, this is kind of a fancy word. It just really means how salty something is. So I just usually say it refers to the saltiness of a substance. And when we're talking about tonicity, there's three types of solutions, and these are always relative to the inside of the cell. That's how we define these words. So here's what the words mean. Hyper means above, iso means the same, and hypo means below, right? Then the word tonic, we said, refers to the saltiness. So something that is a hypertonic solution is a solution of liquid that is more salty than the cell. Okay, because remember these words hyper, iso, and hypo, above, the same, and below, are always in relative to the inside of the cell. Okay, so if I put a cell in a solution of liquid that had the same saltiness as inside the cell, that would be considered an isotonic solution. If I put a cell inside a solution that is less salty than the inside of the cell, then we would call it a hypotonic solution. And what happens to that cell and put into a variety of these solutions varies. Okay, so here's an interesting picture of what would happen. If I put a cell, let's say here's a plant cell, let's say I put it in a hypertonic solution. So all of this fluid surrounding the cell is more salty than the inside of the cell. Well, we said water's the party animal. It always wants to go to the place where that's most salty. So water's actually going to leave the cell, and it's going to diffuse out through osmosis to kind of dilute out that saltiness. So when this happens, water gets pulled out of the cell, and the cell actually shrinks. Okay, so in a plant cell, the cell wall stays the same, but the plasma membrane inside and the cell inside the wall is going to shrink and shrivel up. If I put a cell inside an isotonic solution, nothing happens because there is no concentration gradient. There's the same saltiness inside and outside of the cell. If I put a cell in a hypotonic solution, what happens is that all of this water out here is now less salty, hypotonic, less salty than the inside of the cell. And so water is going to want to actually go into where the party is. Remember, if it's hypotonic, that means the water surrounding the cell is less salty and it's more salty inside the cell. So the water wants to go inside where it's most salty. So water does do that, and it makes the cell swell. You can see that it's kind of getting a little bit bigger as the water is coming in. And so this is, for example, why if you don't water a plant, it shrivels, right? When you don't water a plant, it increases how salty the soil is. And so eventually it becomes kind of hypertonic, and it's going to suck that water out. And when you suck the water out of gazillions of cells of a plant, all of the cells, the water comes out, so then it starts to kind of shrivel and get a little bit weak. The thing that gives plants the ability to stay upright is this thing called turgor pressure. That's the pressure of, filling up a cell full of water. Kind of almost think about it like a whole water bed can hold up a person, so a cell that is full of water can kind of hold up a plant if all of the cells have water in them. Well, in either case, when this becomes hypertonic, then it sucks the water out and then it starts to wilt. But if you were to water this soil again, then it would become more hypotonic, and then water actually is driven back into the plant, and it makes the leaves look nicer. So that's one example of tonicity in real world scenario. Another would be for example, blood. You have to be very careful with blood. And many of you have probably been to the hospital and received an IV where you have that bag of solution that's dripping into your body. They usually do that if they just need to give you some more fluids because of a low blood pressure or just to keep you hydrated. Well, in either case, when they give you that solution, it's got a very specific salt concentration so that it's exactly the same as your blood. What would happen if that IV that was dripping into your body, if that was hypertonic, more salty than the fluid in your blood, then as soon as that hypertonic solution was dripped into your blood, it would suck the water out of the cells because water wants to go from the cell to the place that's more salty, and then that would make your cells shrivel up. And on the flip side, if they dripped just pure water into your body and it wasn't salty, that would mean it was less salty than the cell. And so then all of a sudden that water would be diffusing into your cells and making them burst. Okay, so they have to have a very specific salt concentration that's exactly the same as your blood, so that once that fluid goes into your body, it doesn't affect how big your red blood cells are. It doesn't either shrink them or make them swell. And this stuff is, I'll say this is probably a trickier thing too, so make sure you practice this. And it's going to be so easy to forget where water goes. So ask yourself, hey, ask your neighbor, or better yet, ask your classmate, hey, if I put a cell in a hypertonic solution, what's going to happen? And their response should be, well, water will leave the cell and it will shrink. Then ask them, hey, if I put a cell in a hypotonic solution, what's going to happen? Then your classmate should say, hey, well, water is going to go to the more salty place, and if it's in a hypotonic solution, the more salty place is inside the cell. So water will go inside the cell and the cell will swell. If you can do those things, then you are doing really well. Okay, so that's osmosis. There's one last type of transport which we're going to take a look at here in the next part of this lecture series, but I pointed out is that active refers to the fact that it requires energy. So up to this point, we've talked about all of these different types of ways that stuff can get into and out of a cell. And this is all called passive because it does not require energy. Require energy. Okay. The way that it works is it runs off of concentration gradients. It allows, and it runs off of the normal tendency for things to flow from high concentration to low concentration. But not all of the time are you going to want to move something from a high concentration to a low concentration. Sometimes you're going to want to move something from a low concentration up to a high concentration. When that happens, we call it going up the concentration gradient versus down the concentration gradient. And that is what active transport is it takes a little bit of energy input to pump something from a low concentration to a high concentration, and usually the molecule that's required to make this process happen is called ATP, and we'll learn more about this in the next part of the lecture. So before we end, I've got one last review question here. Can you describe the three types of passive transport? What molecules can diffuse across the membrane and what molecules cannot? Does passive transport require energy? What type of passive transport uses integral membrane proteins? What are the two types of proteins used for this type of transport? Osmosis is blank across a blank. And then lastly, if a liquid has a high temperature, will this speed up diffusion or slow it down? Take a minute or two to answer these questions. All right, here are the answers. For the three types of passive transport, well, we have simple diffusion. Simple diffusion. We have facilitated diffusion. And we have osmosis, which is itself a form of diffusion, just special to water. Okay. What molecules can diffuse across a membrane and what molecules cannot? Well, remember that most of the membrane is hydrophobic, so it's got to be hydrophobic. Or it can be really, really, really small. And examples of these things that are small that can go across the membrane would be like oxygen or CO2, the gases that are continually coming into and out of your blood. Otherwise, hydrophobic, this would be things like cholesterol or fatty acids, those type of things. Okay. Does passive transport require energy? The answer is no. It requires energy, but not of a type that is ATP consumption. It utilizes the potential energy of molecules that want to go from a high concentration to a low concentration. So the answer would be no here. All right, what type of passive transport uses an integral membrane protein, and what type are the two types of proteins used for this? This would be facilitated, diffusion, requires a protein. And the two different types are you have carrier proteins and you also have channel proteins. Channel proteins are just basically passageways that stuff can pass into and out of, and sometimes those passageways are specifically sized or designed to allow specific things through them. For example, there's a channel called an aquaporin, and an aquaporin is a channel that is geared and designed just for water. Okay, so it allows water to pass through, but not other types of things. All right, carriers then are similar, but they require the binding of the substance and then some sort of shape change in order for it to pass through. Osmosis is going to be diffusion of water. Diffusion of water across a selectively, or I should say, not that, a semi-permeable membrane. There has to be a separation in solutes, and if there was a, oh gosh, I'm really losing it here, semi-permeable. Semi-permeable meaning water can pass in between, but not all of the solutes. There has to be a difference in saltiness between the two locations of movement for water in order for the water to be drawn from one location to another. Then if a liquid has a high temperature, will this speed up diffusion or slow it down? It will speed it up because it'll make the molecules move faster. Okay. And then one last question here for you. If I had a cell that was 10% solutes and then the cell is placed in a fluid containing 30% solutes, a couple of questions. A, is the fluid surrounding the cell hypotonic or hypertonic? And B, will water move into the cell or out of the cell? And can you explain why? Take a minute to work on this, pause the video, and then we'll go through the answer. Okay, here's the answer. Here's my cell, and the cell is going to have 10% saltiness. Outside the cell I've got 30% saltiness. Okay, so relative to the inside of the cell, this solution out here, this is more salty than inside the cell, so the word that we use there is hyper, meaning more, tonic. So this word literally means more salty, and it's always in reference to the inside of the cell, so it's more salty than the cell. If I was to keep the cell saltiness the same and maybe reduce this down to, like, let's say 5%, then we would say that the solution is now, relative to the cell, less salty than the inside of the cell, so this would be a hypotonic solution. And if I had it 10%, this would be isotonic solution. But the question, it says 30, so we'll leave it at 30. Hypertonic solution. So now tell me what happens to the water. Well, let's say I've got a water molecule inside the cell. Here's my water molecule right here. Water molecule would look around and look inside the cell and outside the cell and say, which party's got the most solutes? And the answer would be the one that's happening outside of the cell, because there's 30% solutes instead of 10%. So there's more particles, more salts, more solutes in this fluid than inside the cell, so water is going to want to go to that party. Okay. And when I say, keep in mind when I say saltiness, it could refer to things like salt, like sodium chloride, but it could also refer to just a general term for stuff. So more solute could be anything. It could be sugars, it could be protein, amino acids, and whatnot. It's just the amount of stuff that's in a fluid. So water does move out, and then your explanation of why is because water is moving down its concentration gradient. When it's really salty out here, there's not as much room for water. When it's less salty here, there's more room for water, so water is actually more concentrated here and less here, so water moves down its concentration gradient. So here's that same animation I showed you earlier. Go ahead and give it a watch here, and we'll see if we can spot out some of the things that we saw at the beginning but maybe didn't recognize. So here's our nice phospholipid bilayer again, where we've got the fluid hydrophobic region and the hydrophilic tails. This is actually a localized region with a lot of cholesterol molecules in it called a lipid raft. We didn't talk about this. This is a protein that's embedded into the membrane, so we call it a integral protein. And this would be like, these are like little peripheral proteins. Here's a nice integrated integral protein, and if we go back, let me look at if I can go back here once. I want to quick show you something. So this would be an integral protein, and you can see there's a whole bunch of amino acids, and after this changes and folds its shape, there's a whole bunch of amino acids in this long polypeptide, and these would all be a series of hydrophobic ones. And so when you get a series of them like that, it tends to embed itself into the membrane. Okay, so that's an integral protein. This would be a peripheral protein, and that would happen because there's two little small regions that are hydrophobic that allow this to kind of associate with the membrane. By the way, I don't think I ever mentioned this, which is a big error on my behalf, but what these peripheral proteins do is that they can either serve as these can sometimes be enzymes, and so you can put a bunch of enzymes anchor them on the inside of the cell, and they can be like places where proteins are made or destroyed, or not made, but proteins are destroyed, or like amino or carbohydrates are made or destroyed. So that whole dehydration, synthesis, and hydrolysis stuff that we talked about, that can happen in enzymes that are anchored kind of on the periphery of the cell. For whatever reason, this could be a place that localizes and serves as like an attachment point for like different cytoskeletal fibers or other types of protein. It can be kind of a place where it attaches them to the cell membrane. So that's what peripheral proteins would do. But again, most of all of this part of the protein would be hydrophilic, and it would just have a couple of hydrophobic regions that allow it to kind of associate there. And I think that's about it from this animation that we talked about. Some of these things are probably channel proteins that allow stuff to move into and out of. You can see this is kind of like the cytoskeleton anchoring on there. At any rate, that is going to wrap us up for our first part. We'll talk about active transport next time.